Szép jó napot kívánok, schönen guten Nachmittag. You are welcome at the embassy to a very interesting afternoon. Don't worry, I won't speak Hungarian. But I have to praise the speaker of today's event, who is a very famous Hungarian and famous overall in the world, and not only in Hungary. But first of all, it's an event organized by the Dennis-Gabor-Society, which is a very noble troop, which supports the contacts in the field, which for many of us, at least for me, for sure contains a lot of knowledge information where I have no clue what these people are writing, publishing, and talking about. But at the end, it has a very direct impact on the future of the human being. So in that sense, it's also something symbolic that if somebody has intensive and good contacts to Dennis-Gabor-Society, sooner or later has the chance to get the Nobel Prize, which happened some years ago, as we had here the honor to welcome Frau Kariko and Mr. Kraus. Today's speaker is Professor Roska, who is, how to spell it, optogenetic therapy, which frankly when I first heard about, I was totally lost. Now I am a bit more knowledgeable on the issue, but what I learned is first that there is at least 1 billion people who are affected by the eye illness, and how limited the knowledge and the technology which is on the table today can help them. And those pioneers who try to solve one of the biggest problems and challenges for the 100 millions of people is in itself already something which can be very interesting for you. Secondly, as I learned about a survey made in the United States, at least among Americans, the awareness of eye illnesses shows that it's basically for the people the most dangerous, which they are mostly afraid of. And in that sense, how then this problem can be challenged, tackled, kept under control, it's, I guess, something which is well beyond the limits of this room. And my last remark, Professor Roska, you said somewhere that your life is consisting of two parts, 90% of failure and 10% of success, but this 10% is basically motivating enough for the bigger part of your professional life. I am sure that today's event will grant you basically a very useful, successful knowledge level, which will go up to 100%. So in that sense, I wish you a very interesting, challenging afternoon and enjoy both the content and afterwards the hospitality of our embassy. Thank you very much.
So I was not here in the first minute, so maybe the ambassador already mentioned that we are gathering here at a very special occasion. So this is the 125th anniversary of the birth of Gábor Dénes, the Nobel Prize winner, and he is gave us, so we are using his name. And it is a great pleasure and indeed an honor to welcome you all today. This event to introduce you our distinguished speaker, Professor Botond Roska. We are particularly proud that this lecture continues a wonderful tradition. Last year, our symposium on physics was joined by Professor Ferenc Krausz, Nobel Prize laureate in physics in the 2023, whose pioneering work on attosecond laser pulses opened a new window in the motion of electrons. And the year before that, we had the privilege of welcoming Professor Katalin Karikó, a Nobel Prize laureate of Physiology and Medicine 2023, whose discoveries on messenger RNA technology changed the course of modern medicine. To have Professor Roska with us today, another world-leading Hungarian scientist whose work reshapes our understanding of biology and the boundaries of medicine, is both a continuation of this remarkable line and the source of deep inspiration for us all. Professor Botond Roska was born in Budapest in 1969 into an intellectually and artistically vibrant family. He was first trained as a cellist at the Franz Liszt Academy of Music before a hand injury redirected his path toward science, a turn that ultimately brought music's sense of structure, pattern, and harmony into neuroscience. He earned degrees in mathematics from Eötvös Loránd University and medicine from Semmelweis University, graduating summa cum laude. He went to complete a Ph.D. in neurobiology at the University of California, Berkeley, later joining Harvard University as a fellow of the Harvard Society. From early on, Professor Roska's career has been marked by unusual synthesis, combining mathematical precision, medical insight, and a deep curiosity for the neural code of vision. Today, Professor Roska is a director of the Institute of Molecular and Clinical Ophthalmology, Basel, IOB, am I right? That's correct. Yeah, that's correct. Which he has co-founded in 2018. At IOB, he leads one of the world's foremost programs on understanding the retinal neuron network, how it encodes the visual world, and how we might repair when it fails. His laboratory's work bridges basic neuroscience, molecular genetics, and gene therapy. Among the groundbreaking achievements is the use of optogenetic gene therapy to restore partial sight to patients who were completely blind by making remaining retinal neurons light sensitive again. In parallel, his team has developed functional human retinal organoids, effectively miniature retinas in a dish, offering unprecedented insights into both normal vision and disease. For these transformative contributions, Professor Roska has received many of the world's most prestigious scientific honors, including the, excuse me if I don't pronounce this properly, Louis Gentet Prize for Medicine, the Körper European Science Prize, and most recently, the Wolf Prize in Medicine in 2024, which he shared with José Alain Zahel for sight-saving and vision restoration to blind people using optogenetics. However, it is perhaps the scope of his work that makes it extraordinary. It combines curiosity-driven research with real therapeutic hope. It shows us how deep understanding of biology can translate into restoring a lost sense, into giving light back to darkness. So today, we continue our proud tradition of welcoming exceptional scientists whose discoveries transcend disciplines, physics, biomedicine, and now neuro-ophthalmology, each illuminating the unknown in their own way. Please join me in giving a very warm welcome to Professor Botond Roska, and let us look forward to his lecture, which he promises to open new perspective on how we see, both with our eyes and with our minds. Professor Botond Roska will give a presentation on Restoring Vision with Optogenetic Methods. Please.
Thank you very much for the kind introduction. It's really nice to be here. I'm an often visitor to Germany. I have many friends in different parts of Germany. And you probably wonder why I speak English if I live in Basel, which is German speaking. And the answer is I'm horribly untalented in languages. And it was when I was in mathematics, I had a very stupid idea, is that I don't want to represent the same concept with two words in my brain, which makes no sense, but I was young. So I speak the language of mathematics, Hungarian and English, and unfortunately, my German is elementary level, which I'm deeply ashamed, but I have to live with this. What I will talk today about is vision. And as we heard, a very nice introduction is to the concept of my institute when I started it was that if you look at pharmaceutical companies, they usually go, they hear about some molecules, they get some molecules, they want to try it out in humans. And what my institute tries to do differently is that we really start from understanding, and this understanding is the basis of all therapies we build. And this is made possible by gene therapy, where this is really a scientific approach. Every element of what we do is based on understanding, as opposed to testing a million molecules that might work, but you might not know what to do. And so today I like you to a little bit understanding human vision and how fundamentally human vision is different from most animal vision you know of. And when we ask ourselves, why is it such as in the last 20 years we have basically no new therapy? The answer is exactly that, because the human vision is fundamentally different from most mammalian vision, which I'll tell you why. And that poses as a challenge is that how can we understand human vision and how but the mechanics of human vision and how we can use this understanding then to build new therapies. Now, my institute works on 12 different therapies, and I have no time and probably to go through all, so I will talk about 1 out of 12. And the reason I choose this, because this is something I started around 25 years ago when I was a graduate student in Berkeley. This idea was born there. And, you know, I'm a long runner. And I like very much to do things for like 30 years. And things are kind of evolving. And we have the other 11 projects are also going to clinical trials, but this is really the one where I can show you a clinical trial and I can show you how a basic scientist is now the experiment, in a good sense, is really the clinical trial, because it's the only way I know how humans react to something. And then based on what I see in a clinical trial, what the patients tell me, I now build a new therapy. And that's what I like, the essence of what I tell you, that I have an understanding from biology. I try to move forward, but at the end I have to ask the person, what do you see? And from that I go back to the drawing table and try a new one, and this new therapy will be probably in a year from now. But you will listen to this first therapy and see what is the version 2.0. And this is what I like to sort of explain to you, that medicine is not that we build a therapy and then we go home, but is a live experimentation that we know very little about the human body, surprisingly little. I always surprised if a medicine works, you know, because this is not that we understand what we do, and therefore this is an iterative process. How we write a computer algorithm, it's very often an iterative process. This is how we also do medicine. So I study vision. And vision invariably starts with the video. This video, it kind of pours into your eye. And I show you a famous face in Switzerland of Roger Federer's. But the reason I show you, because humans really like to look at faces. And I will tell you what we are quite unique about that. And this video then is, through the optics of the eye, is projected on the retina, which is the back of your eye. And from there, there's a computer which is called retina. I will talk about it. And this computer makes new signals, completely new description of the world, that then is going in somewhere in the middle of the brain. Then it's sent back to the back here, somewhere here. This is a visual cortex. And then information radiates through a large part of the human brain, such that around 1/3 of the human cortex we can measure visual activity. So unlike a lot of animals, humans are incredibly visual. So during evolution, we arrived to this stage, and again, I'll tell you more about why is that so. Vision is an extraordinarily complex task. You kind of take it granted. You just walked in, you are looking at this presentation, but you have now billions of neurons working together in your brain to create an image, what you see here. So the retina is a tiny part of it. So you can ask why I spend much of my time of studying the retina, and there's a very, very clear answer to that most of the diseases of vision are coming from the eye, and the one that we cannot solve medically are coming from the retina. Okay, so if we would be able to treat retinal diseases, we would be able to treat around 95% of all visual problems. So if you are a medical doctor and looking at vision, your key question is that how can I repair the retina? So we can ask, what is the retina doing? And this is my description of around 30 years of research that I have done in this subject, that the retina is an image processing computer. There is one video that pours into your eye, and there is around 30 videos that the retina creates. These videos are simultaneously sent via the optic nerve towards the brain, and they are kind of elementary descriptors of the scene out there. Some of them showing the edges, some of showing motion, some of them showing approaching motion. And so you can think of as you break down the world into 30 components, and these are simultaneously going through the brain. So what is the consequence of that? One consequence that obviously you don't see my face. There's no way you can see my face. What you see is 30 different movies about my face, that your brain concludes something about my face. And I can tell you a lot of experiments that actually you don't see my face. There are many things in my face you don't see, you believe you see, and you don't. And there are many, many psychophysical experiments that can fool you that there are images there that you just don't see. But the point of course, of the retina is not, of this transformation, is not to see everything. The point to say to take away the useless stuff that your brain is not interested. You just need some information that is relevant to you. Now, when we look into this computer, what we call retina, there's a really fascinating structure inside. And I have very little time to explain, so I just say some words about it. First, I told you this is a computer. So many of you believe now this is a computer like this, and it's really not. So what is in this computer? In this computer, there's a lot of transistors, billions of transistors, but every transistor is the same. This is a computer where every kind of transistor is different. So biology fundamentally makes a different computer than our digital computers by, you see these different shapes, these different cells in the retina, and each is a different kind of transistors that are hooked to each other. They all talk to each other and doing computation. So what do they do? First of all, there are cells just simply sampling visual information. It's like a little photodiode. They just measure the intensity of photon at one particular point, and we have a lot of them. So this is basically like a photo camera captures an image or a video. Then at the other end of the retina, there is a cell called the ganglion cell, which forms the optic nerve, and there are many of them, and they transmit the information towards the brain. And in between, there are 100 different cell types organized in a very neat way that processes information that computes these 30 videos. And that's what the retina is doing. Now, the very concept, what I told you, that there are 100 what we call cell type, you can think about 100 different types of transistors, is relatively new to medicine. In biology, when I started in Berkeley, my graduate students, I just basically used the retina to understand neural computation. I had no idea I want to use it in medicine. I was just a curious person, wanted to understand how the brain computes. And the retina was a really fascinating part of the brain because I can peel the retina off, put it in a solution, can show videos, and it computes as if it would be in the animal. So this is an only part of the brain which is fully understandable. That's why I came to study. But at that time, had no connection to medicine. And the connection to medicine really came from my laboratory and several other laboratories when we realized that each of these components, the different transistors, had a different biological composition, so-called gene expression pattern. And from a number of studies, we realized that almost all eye diseases are fundamentally originating from a particular cell type. And this is understandable by studying these different transistors or cell types. This happened around 2010. So suddenly, from a pure academic study of how a part of the brain works, get a fundamental insight that connected to visual diseases, indeed all diseases of vision are cell type specific. And the question was, can we do something to repair then visual diseases? And we have spent many years with a very simple idea, is that there are a number of diseases that originate on these different cell types, and we tried to repair it in animal models, such as mice. We could make specific, genetic mutations, and then we tried to repair. Indeed, we were very successfully repairing in mice. And then come the big cold water. Then we said, okay, now we go to medicine. And we try to translate this knowledge into patients, and we miserably failed. And I like to explain you why we failed and what we had to change, that now we can actually build new medicine in a completely different way than we imagined 15 years ago. So one of the reasons we failed, because the cell types, these different computing elements, are very different in mice and humans. Indeed, very different across species, because every eye is kind of evolved during evolution to be optimal for a visual environment. The mouse has, for example, eyes on the side, so when one is running, the mouse is running, the visual information goes this way. When you are running, your eyes in the front, the visual information goes this way. You run in different speed, in different environment, the mouse. So obviously, during evolution, all the perfection of how you compute these images changed from mouse to humans. And it changed so much, indeed, that the internal composition of these cells changed. So when you repair a mouse, it's fantastic, you become a mouse medical doctor, but it's not going to work for humans. But there is a deeper reason for that. And the deeper reason is that during evolution, humans and monkeys in general, primates, and a new structure appeared in our retina. And this is called the fovea, or how other people know it as macula. You probably heard about macular degeneration. Macula only exists in primates, among mammals. Certain birds have that, but among mammals it's only. And this is responsible for high resolution vision. And this becomes so important for humans, is that if you lose this tiny part of the retina, this is a central part of the retina when high resolution vision is occurring, when you lose it, you report, I am blind. Indeed, we have patients coming into the office and say, sir, I'm blind. By the time you cannot be blind, you just walked in. No, I'm blind. It means that I cannot see faces and I cannot read. So for humans, the most important activity in the time of little computers is basically reading and face recognition. If we lose it, today the biggest problem of blindness is not necessarily we don't see, but we're out of society. So this is part of our social life, and this is what we use. Now, if you lose most other parts of the retina, indeed 95% retina you can lose, and people can have a normal daily life. They have professional life, they can fulfill their job because they have this high resolution part. So to summary, most mammals use vision as an auxiliary to many other senses. Humans use vision as a primary sense today in society to read and recognize faces, but not the whole retina. It's only a tiny, tiny part of that. This is actually the most sensitive part of your entire body. If you lose a millimeter square of your retina, you basically go blind. Now, the brain evolved to look at the fovea. So if you look how much brain tissue is dedicated to a piece of retina, for the macula, you have a massive piece, and you have 1,000 times less when you go to the periphery. So in human evolution, we created this extraordinarily sensitive, tiny, high-resolution camera and put a huge brain behind it to analyze. And we have a very large retina, which we can lose, actually, is not dramatic consequences, with very little brain using them. To demonstrate you that you can see your own fovea in motion, look at this picture, and if you're in the right distance, I think probably many of you have good vision and recognize there are some little barbs if you saw these little lines. You see these little lines? Yes. Do you see that these lines are jumping from one location to another one? Just try to look at it. And you look at one location and you see that it will be jumping this little, don't move your head, just look at this. So if you have good vision, and many of them, what you will see that these little lines, these one you look at, appear at different positions. Indeed, your eye is moving and you have a little high resolution camera, and wherever the eye is moving, you see these little barbs, otherwise you don't. So how you build vision is you have a tiny part of your retina that has high resolution, and you scan the world and put together image. The image is in your brain. It's not there, it's coming from memory. You are scanning it and you're building a picture. Indeed, that happens when you go to a museum and you look at the beautiful Monet picture, you only see a tiny part of this Monet picture. And over time, actually very fast, you build an image in your brain and you believe you see this Monet picture. Now, this is a very special structure, the fovea, I will talk about later, where every little detail will matter. And here is just a picture to tell you that it's a fascinating structure. And a little bit you will have to know about it when I talk about therapies, and that's the following. If you go to the periphery, what you see, there is a hole. That there is a photoreceptor that kind of vertically, there's a vertical arrangement of cells. Now you see the very center, here is a photoreceptor, and it's kind of going side, it's connecting sideways to arrive to the output cells. Okay, this is, before you birth, it's not like that. Before you birth, everything is vertically arranged, and before birth and during birth, this is kind of going on the side. So you create a little hole in the middle of your eye, and the reason is doing this because the light is coming from there, and you can have a very high resolution because the light is directly on those green photoreceptors. So this is just a trick of the optic system, is to make a clear image on the fovea, but it has important consequences for vision restoration. Now, all that from my introduction, what I like to get is that the human retina is fundamentally different from most animals, except of primates. Primates have very similar vision to us. And that poses an enormous challenge to develop therapy. Because I have to develop therapy on something, right? And today I will show you how over the last 10 years, we invented, and our colleagues invented, a new set of technologies that today we can actually build a therapy in a human retina of different sorts. So as I told you that we have 12 different translational projects in my institute, and the one I will talk about, this is actually full blindness. So the patients who come to this have full blindness, mostly from inherited retinal degenerations. but now we are also asking a question that potentially whether this could also work something called geographic atrophy of age-related macular degeneration. In this case, again, important that we are not trying to slow down the loss of vision. We have other programs of that. Here we try to restore in a completely blind person vision that is already lost. You can ask yourself, isn't it easier to slow down the loss of vision as opposed to going to a completely blind person and try to do something? And the answer is no. It's indeed in medicine, this is much easier. And I tell you why. If somebody is blind, I inject something, there's a clinical trial, inject something in the eye of that person cannot be more blind than blind, right? We are really going for full blindness here. That means that the safety concerns are much less than if we go in somebody who has still remaining vision. We have extraordinary care when we go for somebody, both actually who is blind, but if not blind, you know, we really have to argue and argue and argue to set up a clinical trial, because it's the last one we would like to have is to lose more vision that you have. So this is what we call is a real opportunity in medicine to work out technologies that we can later refine to use in cases when we want to slow down the loss of vision. So the story really started in algae, and it started really in Germany by 3 German researchers. One of them is Peter Hygemann, who is in Berlin, who studied green algae. Green algae. And they were just curious about green algae actually can orient itself towards light. They have an elementary vision. They can again swim against light source or towards the light source. And they were basic biologists. They're just interested how on earth this little algae that has one cell can swim towards or against light. And this is actually show how incredibly important basic biology when we just ask how nature works. And so they figured out that this is it does very differently than your eye does it. So your photoreceptor has 20 genes working together in concert to give you a light sensitivity, and this algae has 1. So that's really remarkable. And then you can question, like, why on earth I have 20 and the algae has 1? And there's a very simple explanation, because vision has 2 parts. One is to seeing light, or gray, different shades of light. The second one is able to see across many different light levels. So you don't notice, but when you walked from outside of Berlin inside this room, there is 1,000 times more photons outside than here. Indeed, there are conditions when there's 1,000,000 times or 1,000,000,000 times more photons. So that means that my visual system constantly have to adjust its range of intensities to the one I'm actually in. If you are in your cellar, and I often did it, I did recordings in Berkeley in California, I would go outside when there's 1,000,000,000 times more photon, I would see nothing for a few minutes, and then I would adapt to it. So the algae cannot do it, but it can see some light. So that's why you need 1 gene. But 1 gene generated a huge opportunity, because 1 gene, we can pack them into some viral remainders, some packaging molecules, and we can deliver to cells. So if I would have to deliver today with gene therapy 20 genes, there would be no way to do it, but there are technologies for 1 gene. Now, so the idea that was really born when I was a student is to simply take this gene and put there into somewhere in a blind retina into some cell types that make sense. And this was very dear to me since at that time, since 5 years, every morning I got up at 5 a.m. and I would record from retinas until early afternoon. So I really knew the retina very intimately. There's a lot of days to record from the retina. And it was very clear that there's only 4 ways to do that, to target 4 different possibilities. The details don't matter. What matters here is that there are some of these approaches which you would make the retina light sensitive, but none of the retinal computation would be there, but this would be very simple. What I mean by very simple, that within 15 years you can go to patients. There are others where we had no technology at the time, but we knew could be much better, might give you a real much better vision, but we didn't have any technology. So what we decided to do is that we went ahead for a clinical trial with the simplest approach. We learned what these patients can see, what cannot, and then I will show you how now we go for more complex approaches for better vision. So what we built here is a gene therapy vector, which is very simple to understand. This is a ball. It's a 25 nanometer diameter ball. And within this ball, there is a DNA molecule that we program. So we call them viral vectors, but they're coming from viruses, but it's all artificial. So there's nothing left here, which is coming from viruses. We were inspired by nature. It's all human engineering. And the DNA has 2 parts. One part is just this algae vision molecule. And the second one is a postal address that we kind of invented in my lab and my institute. It's a very fancy postal address, so this virus will enter cells, and this postal address would smell out which cell type they're in and only make the protein if they're in the cell type that we want to be. Now, we are working on it since probably around 15 years on this technology, and we are getting better and better, but we are not yet completely there. So how does it work in practice? We make several thousand of different gene therapy vectors with different postal addresses and different cargoes, and then we have to test them. And then comes the question, I already told you that if we treat mouse, it's not going to work in humans. Indeed, we had a very formal proof of that. We tested many of the gene therapy vectors, and they didn't. So how on earth we get human retinas? And there are really 2 important technologies here. The first is what's called organoid technology, that today we can build thousands of human retinas in a dish in our laboratory. These are light-sensitive, really human retinas. We call organoids, but they are only copies of a newborn retina. So this is a good approximation, but the therapy will go to a 60, 70-year-old person. So it's not everything, but it's a good starting point. So what we do here is that when we build them, they're relatively similar to the normal human retina. And the key point with this is that we can make them mass scale. So recently we did 20,000 human organoids in an experiment. Last year we did another 15,000. So really you can use these human retinas, you can deploy them and test thousands of these gene therapy viruses. And based on this, we can find some that worked very well. And then the question, will they work in a real adult human retina? And then really comes a technology which mostly developed by a Hungarian anatomist, Arnold Szabó, at Semmelweis University. And he, over many, many years, he simply figured out how to keep human retinas alive after the death of people. And this is an incredibly important technology. And it's a technology that nobody could ever discover how to do it in the US or Western Europe. You had to be in Semmelweis University, and I tell you why. The reason is no one would give us funding if we would say, well, look, we will work 20 years on something that we don't know if it works, and it's in a solution, and we might change some parts of the solution until we can keep a human part alive for 30 weeks. They would laugh on us. So why in the Western funding scheme there are a lot of advantages? There are horrible parts of it. For example, it doesn't allow truly open-ended fundamental discoveries such as here, where in an anatomy institute in Semmelweis, Arnold worked for 10 years just changing parts of a solution until he can today keep alive a human retina for 30 weeks post-mortem. This is gold. When I heard about it, we actually met on the streets close to Semmelweis University. I didn't believe him, and he showed me, look, I can keep alive. It was true. So we shipped in all our gene therapy viruses, and we tested on these. And then one by one, we can isolate now real gene therapy vectors that we know they work in the human adult retina. And for somebody who's developing medicine, this is true transformation and it's true gold, since this I can ensure now that this will work in the patient's human retina. It's not a mouse, not a rabbit, not whatnot. This is what we want to treat. So what I like to show is one example now, how we use these technologies now to move ahead and to treat patients, and then what can we achieve with the simplest case. And this is a work together with José Sahel, who was in Paris for a long time, and now he's in Pittsburgh, US, in a small company. We started GenSide Biologics and other collaborators, Serge Pico and Denis Delcara. And what we do here is basically turn the retina into a light sensor. It releases all its capabilities to compute these 30 movies that I showed you, but there will be some sort of an image that is transformed, that is going towards the brain. And this is a very simple therapy. Basically, it takes 5 minutes. It's a standard injection to the eye that ophthalmologists do, hundreds of that, probably in Berlin every day. Thousands of people are injected with what called a VEGF therapy in macular degeneration. And this is a simple injection to the eye. And we had to create a small goggles, which has a video camera watching the world. And it has a projector that is projecting towards the eye, the same projector as you see here, except everything is miniaturized. And the reason we had to do it, because these algae light sensors have specific wavelengths, specific intensities, and we had to do some computation in these goggles. I don't want to go to the details. What I like to show you sort of a snippets how we test this. So these patients are blind, and some of them has bare light sensitivity, some of them have no light sensitivity, but none of them can tell there is an object. So we do a test with or without the goggle, where they have to look for the objects. And the control condition when we take off the goggles, because in this case the room light intensity is not enough to excite these algae light sensors. And then we have other tests, like people have to tell the orientation of bar or other. So I show you a couple of videos, and then the key point is that you look at these videos and sort of conclude what can this patient do and what they cannot. In medicine, wise people who develop therapies always look what the patient cannot do, because it informs us what to be next. So what I will show first, so this was actually our first patient. It's in Paris. And this person has a bare light sensitivity, but no object sensitivity. And we first test it without the goggles. And the task is to say, where is the object? Or to say, is there an object or no object? To locate the object and touch it. So here, one eye is injected, the other eye is pedged. Again, there is no goggles, and the task is to tell whether there is an object on the table or no object. And what the patient is telling, that he sees some light coming from the table, but there is nothing he can see the table. This is typical condition of this stage. And then we put on the goggle, and what the video camera is recording, you can see it on those computer at the bottom. And what we see here is the patient start to horizontally scan the table. And at one point, you can see on the camera already, the object is on the camera. And the person is perceiving it. It's there, he says. It's on the right. And then we ask him to point at the object. This is the pointing part. So first, this therapy, we have a tiny part of the retina is light sensitive. So you can test on yourself. Imagine that you have to find somebody in this room, and you have only a part of the retina. So you put here your hands. This is exactly what kind of vision they have. And then you have to find your friend. So you will imagine that you will have to squint quite a bit to find your friends, right? This is exactly what you have seen, right? So we have seen that this person is kind of scanning, at one point finding the object. Now, what we also see is that once you find the object, imagine your task would be to go to your friend. You would immediately and directly go there. That's what you see. Now, the difference between you and this person, that this person is for 10 years is blind. That means that the information in the brain is completely preserved, that once you see something, how your motor system is working. And this was one of the first lessons we learned here, is that there's an incredible capability of the human brain that going blind, and 10 years or 15 years after, you perfectly have the program in your brain what to do if you would have vision. Okay, that's very, very, very good news for any further therapy that will come in the next 30, 40 years. Now, I like to show another video, basically just variations, so you can appreciate it's the same task, but just important that you have a sort of understanding of how other patients are doing, and indeed how similar they are. So we call patients, he's a patient, now is a task, again, no goggles, and he has to point at it, and we either show an object or no object. So the answer is I don't know, and you will see it again and again, is that if there's no goggles, it just basically means that these patients are really blind. This is very important for us because we have to have a baseline compared to the compared therapy. Here is yet another patient. You can see one is in Pittsburgh, I think another one is in Paris. But if you look at what they are reporting, it's always that I don't know. Or they can't see anything, or they don't know. Now, we put on the goggles, again, in these two other patients. And now, of course, this is, we are tricking the patient. There's nothing. It's also very important for statistics. You see again the scanning motion. That's the strategy, and that's the right strategy of these patients. And now they correctly conclude there is nothing. And now comes the litmus test. And watch again. So the scanning, so the horizontal is scanning exactly what you do with your hands when you do this. And there is your friend. In this case, that's a friend, and there is the object. Okay. So what I'm getting is that you can have one patient. I have many, many patient videos, and all of them tend to have the same response. That means they spend time to search. Once they form an image, they're able to tell where it is. And I have videos of large objects, small objects, this bar, that bar, and probably we can just skip forward and maybe just show you one last video. Is that in some cases, really the precision is extreme. And this is a lady who was 70 years old, at the time she was tested. It might not work. Oh, wait. I think it's now starting. Yeah. So you can see it is a much harder task. She has to actually count pencils and then have to point at one pencil. And what I like to sort of show you is the precision of the pointing. And now she's counting, and now this is a task to point at, and that's the precision of pointing. So that shows that the motor program in some cases is really extremely preserved. So what do we find with 8 patients here is that around half we can see a significant restoration of object sensitivity. The other 4 we did not, and we don't know, is it the injection problem or we don't know where the problem starts. What we see, however, and this was really encouraging, is that we asked, can we predict who can respond and who cannot? And finally, as of 3 weeks ago, we found actually how can we predict. And namely, nothing else matters, just how much we train the person to wear the goggles, not to do the test, but to wear the goggles and a blind person to teach the blind person to look at a projector. And if we teach that person how to the task, you can see the performance goes extremely highly up. So it's not that we have unknown responders and unknown non-responders, it's just they were early in the training program. So this is what we learned, is that they, around 4 to 8 patients, they have very good recognition. It purely depends on the training. Indeed, likely all of them can have good recognition. However, there's no face recognition and no reading. And that's the point which I like to now explain to you, as if you were a scientist in my laboratory and you will fully understand. And I like to understand how then and why we come up with the next therapy. This is actually rather, it took us quite a while to fully understand, but once you understand, it's relatively easy to explain. So I like to go back to the idea which I emphasized to you, that humans are special because we have this area called the fovea. And you, when you see, you mostly use this area. And if I somehow, or somehow you get damaged in your retina, 95% you keep the macula, you will have perfectly good ability to recognize faces and to read. Now, let's look at this structure. And I already showed you that somehow the structure during birth or around birth is fundamentally changing. In the periphery, and I will not fall in the periphery, a photoreceptor is vertically connected to a cell that reports then to the brain. In the center, it's not connected to anything. It has to go to the side to connect to these cells to report something to the brain. Now we do a little bit of mental training. What would happen if we would put a light sensor into these cells that report to the brain? Now, because they are on a ring, so I told you they are kind of shuffled to the side, so they are in a ring. Like this is the ring structure that these ganglion cells are arranged, and I make this ring light sensitive. That's what they have. They have a light sensitive ring in the retina, but the ring has no light sensitivity in the center, so we will see nothing in the center, okay? And because of the wiring of the ring, they will distort the image. This can be just computed in a mathematical code. So they have a very distorted image. All what they can tell is when you scan, you can see the edge of that object and they can report there is something. Now, if we were able to go to the photoreceptors, you would get a real image. So the real realization is that at the site where the high resolution image is formed, it actually does matter a lot which cell type you put. And you have to put in a cell type, your light sensor, which is actually on a regular mosaic. So we actually started to build a new therapy, which was not new because I published it in 2010, the idea. a graduate student. But at that time, we built it for mouse, and we had to build the entire therapy in humans. So now I show you how we develop a therapy in humans in the last few minutes. So all this therapy was done in humans. They didn't see a mouse, they didn't see anything else, using new technology. So as I showed you, when you build a gene therapy, you have this little ball, and you have to engineer, you have a light sensor you have to find, and you have to engineer a postal address. So what we can do now is that we take these human retinas that Arnold Szabó developed how to keep them alive, post-mortem, and then simply add many, many different postal address viral vectors until we find one that exclusively targets the cell types we want, and these called the cone photoreceptors. And here is a bingo. This took 4 years of optimization, where we can, every single human cone we can label, we can bring in a genetic material in the human retina. Now, this is just tells that we can bring a genetic material. It doesn't tell us that it actually, this retina can see again, but we have an enormously powerful tool, namely that if we take a post-mortem human retina after death, this human retina within 1 or 2 days loses its light sensitivity. So it's a general model of blindness. It's losing because it's losing its parts, the light sensitive parts. So what we have actually for 30 weeks is a human retina, but a blind human retina. So this is a perfect model for medicine. So we ask ourselves, can we restore vision to a dead person's retina that it's exactly the same as in the live person it was? And so this is what I'll show you. And the technology goes like this. On the very first day, we have a chip. It's made by ETH Zurich, a chip which has 30,000 electrodes, and I can put a retina on it and record with many electrodes the message that normally the eye sends to the brain. This is electric message that the eye sends to the brain. And then I can show visual stimulation to these retinas. So right after the donation, the death of the person, we take these retinas, put on these chips, and we measure how these cells respond to light. We measure from thousands of cells, and we can have a description of the 5 major classes of ganglion cells, the way they respond. And I just show you one. What we do here is that we flash light. So you can see this light we turn on and then we turn off. And then you can see there are some cells that basically respond when I turn on the light and turn off the light, some of them when I only turn off the light, and a variety of responses. This is the normal repertoire of the human retina, how they respond to light. Now, we know we keep these retinas in culture, and few days later, they become light insensitive, they become blind. And now I come with my gene therapy and pour on my gene therapy and wait 4 weeks. And post-mortem 4 weeks, we go back again and ask, what is the response? How the responses look, which now I generated with my therapy in a blind retina that went blind 4 weeks ago. And what you see is identical. So by now, we can cause in a after death around 5 weeks. We can have light response in human retina that essentially identical to the normal light responses. And this is how we know that it's time now go back to patients, because this is significantly much better than our first therapy. We started a small company on that technology, which probably will be in a clinical trial in a year or year and a half. So there was one question, of course, is that how do we know that there are remaining photoreceptors in a blind person? And this was an absolutely crucial and critical question, since when I was in medical school, what we learned is called structure-function correlation, namely, if you have no function, you have no structure. So blind patients have no photoreceptors. But one thing that I learned as a researcher is that what you learn in university is 50% true and 50% is false, but you don't know which 50%. So we started to, I went around in ophthalmologists and asked them, how do you know? What's your evidence? And within a year, it turns out there's no evidence. An old professor told them something, and the old professor to the young professor, and it went generation to generation. So we had to do real science out of storytelling. And what we did is that we went around the world to around 15 different countries, including many European countries, US, China. And we can directly ask that in living blind patient, because we have an ophthalmological tool called optical coherence tomography, where we can quantify the remaining cone photoreceptors. And what we learned from these studies around the number of countries, and this is the only science slide you have to look at, so I described to you, is that what you see here is the volume of the photoreceptors in the center of your retina. And what you see here is a visual acuity, how well you see. And the blue is normal people. You can see that some people have less photoreceptors, but they can equally see well. So these numbers, increasing the number, 3 is blind, 0 is perfect vision. You can see that some people have a lot of photoreceptors, and some of them has very little, but they can see the same. However, when you look at blind patients, you can see that there are some patients completely blind who have as many cone photoreceptors as normal, and some that has less. So that means at least 50 or 60% of blind patients do have remaining photoreceptors. Therefore, the therapy we developed in postmortem human eyes is valid for that patient population. So I'd like to briefly finish my talk with a completely different thought that I was telling at the beginning is that the way my institute looks at therapy is that therapy is continuous with basic understanding. So we really always look how biology works, and from this working, we designed a new therapy. But this looks a very unilateral process. It looks like we understand the brain, and there we make therapies through brain. I like to show you that indeed by building therapies, you can suddenly find completely new biology by just using the new technologies we built. What do I mean by that? I showed you that 3 weeks after death, we have a part of our brain, the retina, which is a computer, which is fully functional in a dish. I restored vision to the cone photoreceptors. And this is a completely new reality that in the laboratory, we have now a functional brain part, post-mortem, that we can study for weeks. We can finally go and understand how the human retina is working. We understand how diseases, actually we can program in diseases into this normal human retina. I can program in genetic diseases with some technologies we develop. So finally we are in a realm that we don't necessarily have to study just animal models, but we can also look at human models. We can build disease models, and we can model the real structure that we would like to repair. And this is just showing some recording of the human retina around 9 weeks after death. It's fully computing how it does. Now with this, I like to finish with thanking for a lot of people who contributed. My lab, my current lab member, it's actually larger, but I always forget to put some new people. They are constantly coming. And a big thank goes to José Sahel, with whom I work together on the ganglion cell-based therapy that is in clinical trials. And with Péter Györkös, I like to say some words about it. Bence is a Hungarian who was at Harvard when I heard about him, and he's building gene therapies for the ear. And Bence figured out that the eye is much more interesting for gene therapy, so he came to Basel, started the residency, and I talked to him every week. And I told him that, there are a lot of medical doctors, but very few people who are medical doctors and understand research. And in 1 year of hard work, I convinced him to leave medicine and come to my institute as a head of translation. And this is really with whom he is a true expert of translating gene therapies, and with whom we are developing this second generation of therapy that we hope to restore now form vision, or the ability to see faces, not only to find objects. And with this, thank you very much for your attention, and very happy to respond to questions.
So maybe I will go with the microphone and put this under microphone.
This one, yeah.
Okay, so there's a microphone. Who wants to?
Hello, thank you. I have a simple question, maybe it's stupid, but the patients I saw there, they could hear that the object was put on the table.
Yeah, they're not. So this is really, it's done in a way that either there's noise in the room or they have nothing. So this is tested many, many times before. So you see here that there are months of testing to make sure, for example, that there's nothing they can hear. And so there are a lot of controls. It's all video recorded, and everything is analyzed through sound recording. So there is no way they can tell by.
Okay, second question. They could see the object, how we could see it on the screen?
No.
No, okay.
No, definitely not. So
what they're seeing?
Yeah. So what is projected to them is actually not what you see on the screen. What is projected to them is at every pixel, if there's a change of intensity, there's a flash of light. So when the patient is not moving its head, there is no visual input. When it's moving its head, then of course at the edges there are flashes that going into there. So it's kind of a motion detection. Every little pixel there is a motion detector that you would find in your cellar when you go down. So and that's what they are flashing, and that's what they are seeing. Yeah.
I'm Detlef Ganten. I would like to come back to the very beginning of your talk. I'm not in ophthalmology at all, but I'm trying to understand science. I think one of the fascinations, as you said in the very beginning, is the complexity of the eye and the various mechanisms, brain mechanisms behind giving all the pictures, which we then see or interpret or whatever. There's another dimension which I'm very interested in, and that is the future challenge of mankind, if I may say so, is understanding complexity in various areas, geopolitics and science and human interaction. So this complexity is hard to describe because it's so broad, and nobody understands the details of this complexity. So what do we need is pictures of complexity. And I think this is, to me, the very interesting part, in addition to gene therapy, of the eye and your orientation with research. Use the eye and the pictures we produce, mostly individual pictures, of complexity which surrounds us. And understand these molecular mechanisms, I think, will be very important to convey the importance of pictures for communication.
I understand. I think it's a really nice thought, and in mathematics I was fascinated with complexity, and we studied Kolmogorov complexity and other kind of mathematical ways. My father was a mathematician, also was very interested, so I spent actually a lot of time of formalizing. It is true that probably for humans, the biggest complexity we can is with pictures and how we deal in a complex world. Imagine that you are running and there is a flow into your eye and you're capable of making decisions at that speed, or a dog you see that is unimaginable today, even with AI vision. We are nowhere there to that. So I think it's a very nice idea. The retina itself, that we almost fully can understand by simply partial differential equations. What we don't understand, the true complexity comes after the retina. And there we get actually completely lost. We don't know why you need 1 billion neuron to create vision. There's no reason for that to be. And as I said, it's a very nice idea, what you raised. Yeah, thank you.
Okay. So, question?
Thank you very, very much for your nice presentation, and sorry for my naive question. I have two questions. The first one is that when you were talking about blind people, do you also mean the people who are born blind and they don't understand the function of the brain?
Yeah.
And the second question is that, is there a threshold for this photoreceptor to be the number of photoreceptor that the patient can be still, you can work with?
Yeah. Very good question. So the vast majority of blindness is acquired blindness. Even if you have a mutation, genetic mutation, very few kids are born blind. There are some. Of which who are born blind, there are some which has no optic nerve. And in this case, this therapy would not work. We'd need an optic work. Those that born blind, like in some Usher syndrome cases, or early Usher 1 type, we don't know the answer. And the likely answer is probably we can help them, but they would get less visual activity back as somebody who had vision. And the reason I'm thinking is, as I was once I spent some time in a Chinese hospital where they have been looking at patients from poor areas who had congenital cataract. It just happened to be, it was a Sunday, my talk was there, and there was a girl whose cataract was removed and she never saw fully in her life. And this girl was able, 10 minutes after coming out, able to navigate in a complex scenario. It doesn't mean that she could recognize faces, but had some visual activity that was clearly incomparable to what before. So from that, I concluded it's probably for this born blind, they will regain some visual, but how much is probably, we will have to see. Now, I had to mention is that, this field was a funny field because when we first reported using algae for restoring vision, everybody laughed on us. I remember in a conference that they pointed at my poster that there is this guy who lost his mind. And today there are probably even 20 companies around the world who's having optometric visual restoration across all. We have this company for cones. There are 10 companies for ganglion cells and a number now for bipolar cells. So I think that we very soon will know this. Now, my prediction is that this is still early times. As you see, this is still experimental clinic. And what I hope is in the next sort of 10 years, it gets refined to something that could be usable actually for patients who go to the doctor. So it's very important. This is clinical trials and not yet. Now, your second question was?
The number of photoreceptors, is there a threshold?
Yes. So it's a bit more complex question. I have to give you a picture. In the fovea, normally in your retina, there's one layer of photoreceptors, cone photoreceptors. I didn't talk about the rods, but cone photoreceptors. In the very center, in the macula, there are 6 layers of that. So that our calculation that until at least one layer is there, this would work. That would put around 1/6 of the photoreceptors. But how it pays off, we don't know. As usual, clinical trials will start with people who have maximum, and then they move back. And vision is graded in a sense, is that probably it's just the capability will be less for those who have less photoreceptors. But we will see from the trials. Yeah.
Okay, so I think there is one.
Aber ich nur auf Deutsch kann. Mein Englisch wahrscheinlich noch schlimmer als Ihre Deutsch. Also, meine Frage an Sie. Diese Ihre Entwicklungen mit Augen und mit diesen Photorezeptoren und Nervenrezeptoren, kann man doch auf Basis von diesen Entwicklungen auch wissen, wo Demenz sein könnte, um das irgendwie voraus zu wissen und dann stoppen oder...
Yeah. If I understand, you're asking about dementia, whether this can be... So, it's a very interesting question. Directly not. So I doubt that optogenetics can be directly used in dementia. But the way we study these neurons and the technologies we and many others build likely will be useful also for building long-term therapies for dementia. You probably know for Alzheimer's disease there are currently people injecting antibodies, which is not very effective, but is a very first step. And potentially one could use gene therapy in order to avoid repeated injections in dementia cases. So I think that the answer is that technologies can be used, like gene therapy, but the details, we don't understand dementia, where is it coming from, and until we don't understand, it will be difficult to build these therapies. It will come, and I hope that some of these technologies will be useful, but not a one-to-one translation.
Okay, there is one question.
So I have a very simple question. It's like the patient was in a room and he was able to recognize the object that was the one by motion recognition and motion movement of the goggle. So what if he is just outside the room and there are the several objects? What will be its frequency to?
Yeah, so of course we have a real life experience, and we do have these questionnaires, and that really depends on the person. One person, for example, at home. So first of all, this is not useful outside. If you go out a busy street, this is not useful. You will be completely confused. Where it became useful, for example, one patient started to cook using this because it was a simple environment. He could recognize clear contrast. So in its current form, it just shows that to restore a certain amount in a very simple visual environment can give you some object sensitivity. At home, there might be some use, but I think I look at more is that the step forward, based on we can build the next round of therapy to get something that is useful. So think more about this as a scientific experiment, not as a final treatment.
Okay. So maybe I ask the last question.
There is a question.
There is one.
Yeah.
Who?
Thank you. My name is Nishik. I'm from the Protestant Blind Service, and I work also with some people who have RP or ASHA, or this macular degeneration.
Yeah.
And my question is, if you are trying this therapy, which is very very impressive, has this therapy be done repeatedly because the foundation of the sickness is still there in the body, or I mean, have the cells given repeatedly?
Yes, very, very good question. This is a single therapy. You don't need more than one injection because this genetic material, it builds into the ganglion cell and it stays there for a very long time. So that's a difference between in macular degeneration. Usually you hear about injection of Eylea or Lucentis or another, which is a monthly. Those are proteins. And here the protein is made by a DNA which stays in the nucleus of the cell.
Okay, then if there is no more question, I have the last one. It is a bit technical. So because here in Berlin we are so much about the organoids, please explain us what's the advantage and the disadvantage of that organoids. What can you use for, and what are the limitations compared to this magic Semmelweis technology?
So organoid we have to think about bit as a mouse model. So it's human cells, but they don't have the proper organization of the retina. They're very young. They are missing the immune system. We can, of course, put them in. They don't have microglial cells. They don't have vasculature. Many diseases is an interaction between blood flow and the cells working together. So one way to think about, and I showed here, we have like 5 different types of organoids. We can also make now sclera, choroid, all sorts of organoids as others. And I think to me, it has 2 uses. One is that we can mass produce it, and we can do some preliminary screening. But we never can get a final answer because it's not an adult tissue. So, or for example, we have a program that Bence György leads on Stargardt. It's a gene editing program for Stargardt disease, where from a patient we grew with a normal mutation, and so you can edit the normal mutation, and we could test 1,000 conditions to get a good editor, and then we test in a human. So it's in other words, we have a lot of tissues, but not real human. Adult, which we can do as a pre-test. That's how we use it.
Okay, thank you very much. So I think that was the last question to Professor Roska. So let's give a big thank for this fantastic presentation. Yeah, thank you so much. And now the technical problems hopefully solved, so we could have László Bódis presentation, and after that there will be a reception, so you are welcome to participate. So you are between the...
Thank you so much once again, and sorry for being late from this very important conference. Actually, my flight from Budapest, on the flight there was somebody got sick and we have to go to Dresden first, and after that we were able to come to Berlin, but fortunately, he's okay, and everything was fine. So my very short presentation is about how we in Hungary are working on developing the Hungarian deep tech ecosystem, and I think that the example that Professor Roska mentioned to you about how they're working on restoring human vision is a very good example, and I will talk about the systematic approach that we are doing in Hungary. But first of all, what is deep tech? It has, I'm using, it's not my personal definition, but a definition of an international company that defines deep tech as a novel scientific or engineering breakthroughs that are making their way into products and companies for the first time, which is very important. That so basically there you have an engineering or scientific breakthrough, a new research solution, and the other very important part is that these are coming into companies for the first time, and these technologies or products are also bringing solutions to very important societal challenges, which is very important, such as physical and mental health, as we heard, or clean energy transition, cybersecurity, or resource scarcity, all of those very important societal challenges that all of our nations are facing. So if you look at the last 10 or 15 years of innovation, most of the new innovative technologies were in software and digital technologies, but what we would like to see more and more to bring new technologies that are very important for our societies. But these are not just important for our societies, but these can make very significant economic competitiveness increase through creating new high-tech companies. So there are several subsectors which we consider deep techs, such as AI or the future of computing, novel energies, space tech, and different advanced materials, but also life sciences as new therapies, what we heard, or new pharmaceutical solutions or new medical devices are also deep tech companies that has 5 main defining factors. First, as I already mentioned, the new scientific or engineering breakthrough. The second is these technologies has a global scope. They have very high R&D intensity, as we heard. Professor Roska mentioned, that these new therapies, there's still decades. We still need decades until these can be applied as everyday in our everyday life. Obviously, the founders have very high expertise in the given technological field, and the fifth is also very important. These have very significant financing needs. How do we approach the scaling of this ecosystem? In Hungary, we have 3 technological areas where deep tech companies have already succeeded. The first is absolutely life sciences. We have seen some in the last decade which were very good successes. And we have another area which is big data and analytics. Most, I would say, the technologies that are based on mathematics. And the third topic where Hungarian startup companies have good success stories already is fintech and cybersecurity. And when we are thinking about enhancing this whole ecosystem, we reckon there are 5 key enablers of a successful deep tech ecosystem. The first is the business environment where all of the companies are operating. I will get back to these factors in detail in the coming slides. The second is talents. Professor Roska mentioned Péter Györkös, who he were able to get on board. It took a year, so talents is obviously very important. The second is academia, research institutions and universities that are conducting the research. The fourth factor is funding. In funding, I mean government and private funding as well. And the fifth very important factor is corporates who can bring these solutions to the markets. Let's see how Hungary is developing in all these areas. So first, business environment. Maybe a lot of you have already heard about the World Bank Group's Business Ready Index, which is published each year. Previously, it was called Doing Business. Since 2023, it's called Business Ready, and it compares more than 100 economies through 3 pillars. The first is regulatory framework, the second is public services, and the third is operational efficiency. And as you can see, Hungary's rank is actually globally first among the regulatory framework all around the world. In public services, Hungary's rank is 5. And the operational efficiency is 14, so that means that Hungary has a very competitive business environment already. We provide very significant tax breaks for conducting research and development activities in Hungary. We have the lowest corporate tax rate in the whole European Union, which is with 9%, and we also provide different cash grants for those companies that are bringing R&D centers and R&D investments to Hungary. And besides that, we are focusing on changing the different regulations in order to help our startup ecosystem. We introduced last year one of Europe's, I would say, one of the most favorable ESOP regulations, which is how you can motivate your talents, basically. The second is a convertible note regulation, which is also a funding structure for startups. And we provide tax-free IP port possibilities to businesses. So I would say the business environment is pretty good in Hungary. The second very important factor is talent. I just brought here some key figures on different aspects. First, that obviously in different technologies, the ICT knowledge is very important. And as you can see, 10 years ago, if we look at the student entrance in bachelor programs in Hungary, 4.4% of the students picked ICT studies in bachelor programs. Now it's more than 12%. Last year, the most students were enrolled in ICT programs. This was the most frequent field of study. The second is the PhD programs and the number of PhD students. As you can see, in the last 10 years, the number of PhD students who are enrolled in the programs has been increasing more than 50% in Hungary. And what is also very important, then the ratio of foreign PhD students, the students who are coming from abroad in Hungarian PhD programs, has been increasing by fourfold, from 7% to almost 30% in Hungary. And besides the technological talent, we are focusing also on the entrepreneurial talent. How can we help our young students in the universities to not just be good researchers, good scientists, but also get to learn the knowledge that is important to create startup companies. And we have more than 20,000 students who were already enrolled in different entrepreneurial courses, and from this year on, we provide a specific entrepreneurship course for PhD students as well. The third factor, as I mentioned, for a successful ecosystem was academia. In Hungary, most of the research are conducted, besides the enterprises, of course, in the Hungarian Research Network, which contains 14 different research sectors in STEM and life sciences, and we have the university ecosystem. And 5 years ago, in 2019, I brought you some key figures. So the number of universities who were among the top 5% globally in Hungary was 7. The number of patent applications in all the academia, so in the research institutions and in the universities, was 25, which is a very, very, very low number. And the research and development spending in euros were 400 million. Last year, we had 12 universities among the top 5% globally. The number of patent applications has been increased by a factor of 6. And the R&D spending of academia has been more than, it has been increased by more than 50%, which is, I would say, the Hungarian universities and research institutions are on a good track, but what we are working currently on very heavily is how can we translate these research outputs, as Professor Roska mentioned, into innovative companies, into startup companies. And that's why we restructure the technology transfer processes within the universities and research institutions. We are building science parks around our universities, and we are just started to establish new deep tech venture capital funds in Hungary. Fourth factor was funding. On this chart, you can see the increase of the Hungarian GDP in this green circles, and the R&D spending. You can see that it has been significantly increasing. As a percentage of GDP, the Hungarian R&D spending currently stands at 1.4%, and our goal is to increase it with all of the other European Union member states to 3% until 2030. And we created a support system that are helping startup companies, starting from the innovative idea, or I would say the research result, until where market funding, venture capital mainly, can play a decisive role. And we just today launched 2 new programs, which we call Life Sciences Catalyst Program, which aims to help innovative research results to translate into therapies, into new medications or medical devices. And we are establishing new deep tech venture capital funds and a life sciences venture capital fund in Hungary that are focusing on these technologies and funding these technologies. And besides that, obviously, there is a very and increasingly vibrant, I would say, venture capital ecosystem and in Hungary, with several incubators and accelerators, local and international venture capital funds, and different supporting organizations. And the fifth factor are the corporates, as I mentioned. In Hungary, the amount of R&D spending has been increasing significantly, as I mentioned, and partially this is coming from, besides the government, from the multinational companies who are establishing new research and development centers in Hungary, and we're establishing in the last decade, mainly in 3 sectors: automotive, IT, and life sciences. And as you can see, 10 years ago, the number of research and development personnel, so researchers and developers who are working in the business sector was around 22,000 people, and the R&D spending of the enterprise sector was 800 million euros. Last year in 2024, the number of R&D personnel has been 36,000, which is, again, a 60% increase in 10 years. And the R&D spending of the corporate sector has been increasing more than 2.5 times in the last 10 years. And as you can see, there are some, I highlighted some of the most important international companies who have research and development centers in Hungary. And good news that just brought you an example why this is also very important, because those young talents who are working in multinational environment, in multinational research and development centers, now starting to establish new startup companies. I brought you an example from Bosch, which is, you all know, a very important automotive tier one supplier operating in Hungary, and has a very significant research and development centers with more than 3,000 developers, engineers. And two guys just established a new company focusing on autonomous driving, and they were able to get a 3.5 million pre-seed funding in the Silicon Valley already, which is a very good news for Hungary. And we initiated a new model in order to bring the most important corporates working together with their universities. I would say the old model, which is not a bad model, but the old model was we funded the universities to bring in new corporates that they can cooperate with. And now we, I would say, reverse that. And we are now funding and motivating big corporates to pick those research groups in the different institutions, different research institutions, different universities, that they can be working together on different, very exact development programs. And we were able to finance 6 Hungarian corporates, mostly, I would say, in life sciences, in medical devices and pharma sector, to work together with the universities and bring together new solutions to the markets. Obviously, I mentioned some key results, which are very important, but we still have very important challenges to solve. First is, I would say, not a Hungarian problem especially, but I would say a problem in the whole world, especially in Europe, is the entrepreneurial mindset in academia, which is very low still in Hungary. That's why I mentioned we are now helping PhD students with different entrepreneurial courses in order to increase this mindset in academia. The venture capital funds that are available for deep tech companies are still very low. I would say this is also not just a Hungarian challenge, but a European Union level challenge. As you may know, 52% of the venture capital funds are raised in the United States, 40% are raised in China, and those only 5% of the venture capital funds is raised in Europe. These numbers, these figures are coming from the Draghi report, which was published by Mario Draghi 1 year ago. And third is the international visibility of our ecosystem, which we have to work on, because innovation is an international business. So with that, I think the example that Professor Roska showed us, how can we restore the vision of human, can be a very good example on why is the deep tech ecosystem in Hungary or in the broader European Union is very important, once again, because this can bring solutions to real societal challenges. Thank you very much.
Thank you very much for this interesting presentation. Any questions to this? Yeah, so I think there is one maybe interesting debate between the academic science and the translational science, that how much academic science you need, and really those researchers who are the academic researchers would be those that will establish the startup companies. But I think this is just and my. Maybe you want to comment on that.
I think Professor Roska is much more should be answering this question, because basically what he does is a good solution to this question. And when we talked about it, but he will correct me if I'm wrong, is that obviously you have to have fundamental researchers in your group, and you have to have those engineers who know how to translate the fundamental research outputs into new technologies and new therapies in his laboratory. So it's not that we want every fundamental researchers to create new spin-off companies. Obviously, that's not what we would like to see, but we would like to see that our universities and our research institutions mainly should focus on bringing new technologies into the markets, because that's the only way where we can create impact for our society and for our economy. So that's what we have to structure very well in our university ecosystem. And we also discussed it with Professor Roska, mainly most in our Hungarian research network, because it's not always easy to do world-leading research in universities, but that's another question and another debate.
Thank you very much. So I think that then I close today's session. So I think next year we will we were talking about what could be a nice, interesting topic to talk about, and it was exactly this, that just to show some examples that how basic science can be translated, and we will introduce some interesting examples. So I thank you all for coming, and enjoy. We have some reception outside. Thank you very much for coming.